I'm Jonah Comstock, Editor-in-Chief at Pharma Forum. I'm here at ASCO 24 in Chicago, and I'm joined by Chris Haqq. Chris is the Chief Medical Officer of Elicio Biosciences. Chris, thanks for joining me today.
Yeah, thanks so much, Jonah.
So I want to hear about I'm very interested in your data, but let's start with the company, because I'm not familiar with Elicio. So you guys are maybe a smaller biotech. Tell me a little bit about you. Tell me the story.
Yeah, so Elicio formed around a spin-out of technology from Darryl Irvine's lab at MIT. And the whole concept is to make vaccination better by engaging the lymph node. And I like to think of that as the brain center of the immune response, because the critical interactions between the antigen-presenting dendritic cells and T cells happen right there. So getting the key players all in the right location to engage and to make the responses happen the way the patients need for an immune attack on their cancer is what we're formed to champion.
So your data this time around, it's a cancer vaccine, but it sounds really interesting because it's an off-the-shelf vaccine with a personal response. So tell me about that.
Yeah, that was a sort of unexpected finding. We always have conceived that targeting shared neoantigen, which is what we've done with our lead program, it's targeting KRAS, which is present in about a quarter of solid cancers out there. So we thought, well, that's targeting the driver oncogene, which is a good idea because the tumor can never shed that and lose that antigen to escape the immune attack. So that's the advantage of it. But on the downside, you might not be able to get to participate in that immune response. The individual patient's changes that just happen in their own tumor that are not shared among that person and any other patient. So we asked the question with our immune responses, do we see actually the T cells that are specific to RAS? And yes, we do see those. In fact, in the Nature Medicine publication in January, that was in 84% of the patients. We're up now in this new trial that we've just presented today at ASCO to 100% of patients. So really happy with the dose finding that we've gotten to the right range now where we can confer a response in every patient who's taken the drug in our trial. And what we then built upon is to ask about this question of the personalized antigens, this phenomenon that's called antigen spreading. So to interrogate that, we looked in the sequence of the patient's tumor and found mutations that are particular to them, that are just in their own cancer and not in any other patient. And we made peptides that are able to allow us to ask the question, did the immune response spread to be able to target those other antigens? And sure enough, we found that in all of the patients, the 6 patients at the high dose where we did this assay, we saw all of them have this antigen spreading response where they've brought in the personalized part of the response to what's going on with the immune system. So that's super exciting. We weren't expecting it to be as high a proportion of patients that engage that aspect of immunity, and it really suggests that there's a great promise to this line of investigation, because by having that multi-targeted approach, it may help patients to have a more durable effect from the administration of this type of therapy.
So tell me about the intervention itself. What cancers are we in here, and what's the expected outcome? I know when we talk about vaccines and cancer, we're not talking the way we talk about vaccines and infectious diseases. You're not giving it to everybody. It's more of a treatment, right?
Yes, so KRAS mutations are present in a high proportion of patients with pancreatic cancer, around 90%, and about half of patients with colon cancer, about 30% of lung, and several other tumor types as well. So we've begun our trials in patients with pancreatic and colorectal cancers. That was the data published in Nature Medicine in January. And the present trial that we reported today, 13 of the 14 patients had pancreatic cancer. So this is a great area to investigate because there's so much unmet need in the past. Patients that are similar to the ones we enrolled in our trial. These are patients who have had surgery and chemotherapy already, but have a persistent tumor DNA that's detected in the blood or rising tumor markers like CA19-9. Those patients have had only about a 5-month time from the time of surgery until they have a radiographic relapse and need for the therapy for their cancer. So our idea here with this study is to go in that window of opportunity before there's a relapse. And that will give the immune system the chance to be able to maximize the ratio of the T cells to the microscopic nest of tumor cells, and hopefully confer a really long period before they might have a radiographic relapse and need further treatment. So that's the idea, and we're seeing the responses. We saw that 70% of the patients had a reduction in their tumor marker, either the circulating tumor DNA or the CA19-9 in our cohort. So that's pretty nice to see in a phase 1.
Right. Now, this is relatively early data, so
Yes.
it's going to be a few more trials before you're ready to really, I guess, move it in towards the market. What does the road look like from here?
Yeah, so we are continuing to follow the patients that have participated in this phase 1 long term, and so we'll be able to take a look at their time until a radiographic relapse. So that's the endpoint that we call the disease-free survival time. But in addition, we've begun a randomized phase 2 that will provide definitive data where we can compare the group that has received the vaccine. It's a 2-to-1 ratio, so 90 patients will receive the vaccine ELIO2 up front, and 45 patients will initially be randomized to be observed. And then a nice feature of the study is that the patients initially observed can elect to cross over and receive ELIO2 at the time that they do have a radiographic progression. So in this one trial, we'll learn about the activity in this adjuvant setting, that is where patients have had surgery and chemo up front. But also in the group that elect to cross over, we'll learn about the activity in the metastatic group, which is another area of huge unmet need in pancreatic cancer. So we're excited to take this next step with the program, and to get that randomized data will be very important to bringing it a closer step to the product approval. In the past, several approvals have been given for adjuvant treatment on the basis of the same endpoint, the disease-free survival, that we'll use for this study.
So how does your work sort of fit into the larger landscape of oncology right now, on the different avenues for innovation? What's the role that these vaccines are going to have to play in the long term?
Yeah, so we think of it as the dawn of a new era of therapy for many of these solid cancers. It's been useful for many liquid tumors to bring immune therapy into the forefront. For example, with CAR Ts for CD19 lymphomas. And now we're starting to see that pancreatic cancer and other tumors that we thought might not respond well because they're considered a cold tumor, not many T cells found when we look on a biopsy. We're starting to find now that when we generate the T cells that are needed to recognize a tumor antigen like KRAS, they will start to have an effect that we can measure now in our trials. So it's an exciting time for the field. We think that there's a lot to be done with our platform and others that can produce large T cell responses like this to confer benefit. And a lot more work needs to be done in further trials to do the combinations that will be needed to see how we integrate this with the standard approaches that are used now for therapy in these solid cancers. There could be combinations with chemotherapy, combinations with small molecule therapeutics, and even immune-immune combinations. We could combine a vaccine like we're making with adoptive therapy approaches, making engineered T cells, and there's a whole variety of possibilities now of how to move towards the future. And I believe that's an important part of our future because in my career I've seen that we started with many single agents that were active but not able to provide durable benefit, and then by combining them into regimens like CHOP chemotherapy for lymphoma, then we can eventually get to a stage where most patients end up with a durable remission, and some patients even can be cured by the combination approaches. So we're very early with integrating the immune approach in solid cancer, but it's nice to see these initial results showing us that we can have some proof of concept from the early data, and we're looking to do the definitive studies now that will bring this forward so that we can integrate it as part of the mainstay of treatment for patients.
Awesome. I've been asking everybody this question. It's not a newsy question, but what advice would you have for a young person just starting out in the oncology field right now?
Yeah, that's a great question, and I'm always asked when I begin working with any colleague if I've asked them to tackle the worst project. And I think the answer that I would give is to not trust only what you see in mice. I think, of course, we have to look to the animal models to give us the confidence that we need to take an approach now into human patients. But I always tell my colleagues that they'll be the ones who tell me if I gave them the right project or not, because it's the human data that is really the most important. And I think it's very important to remember that mice and humans have quite some differences, and that there can be big surprises. I've seen before in my career that drugs that looked a little bit too toxic in mice ended up being quite effective and well tolerated in the human trials. For example, abiraterone, which I worked on in the past. So there's a lot to be said for doing the translational work to take that first step in the clinic. And I would encourage everyone to take molecules forward that have a sound scientific hypothesis, and to ask the human patient experience to tell us the best value and the best place to use those therapies. It's a great area with a lot of need. It's a great career, really satisfying. So I'd encourage every young investigator to get involved in clinical trials. They're really important to inform us about how we can move the whole field forward.
I love it. What else is on your mind? What have you seen or heard already? We're getting towards the end of Saturday. What's ASCO like for a person like you, coming from maybe a smaller, not one of the big pharmas, or biotech?
Yeah, it's always exciting for me to come to ASCO. I've been coming since I was an undergraduate student working on DNA repair in a basic science laboratory, and I felt very comfortable at ASCO because it's really that meeting of the minds where you get the translation happening right in front of your eyes. You can see some preclinical results, lots of phase 1 results, and then you can learn the strategy of how to design a trial to take things forward and get the results that are needed to bring it out to the benefit of patients everywhere. So I enjoy coming to ASCO. One of the things that's really caught my eye at this meeting is the beginnings of how we'll use artificial intelligence to inform the field. And I guess with teenage kids that know a lot more than me and are much more tech savvy, so that's definitely something I feel like I can learn from at this meeting. And I'm very intrigued to see how we'll be able to leverage the tools from AI as we move the whole field forward.
Fantastic. Thanks so much, Chris. Really great chatting with you.
Yes, very nice to speak with you.
And good luck.